Hi everyone, welcome back to the second video for Chapter 1. Just as you did for Section 1, keep in mind these learning objectives, and you want to be able to answer these by the end of the section. So once we were able to see microbes with the help of these microscopes, we began to realize how diverse they were. So we had tons of microbes that differed in size, they had different shapes and different reproductive rates. We needed a way to organize all of these new living organisms. And that's what taxonomy is. Taxonomy is the scientific method of classifying, naming, and identifying organisms. And this could be grouping them by what they look like, what sizes they are. It could be physical characteristics such as those, or later on we'll organize microbes based on their metabolism. Linnaeus is often known as the father of modern taxonomy, as he created the earliest system of organizing or classifying these organisms. He created the standardized system in order to have scientists be able to discuss organisms using some kind of consistent format or consistent terminology. He published his work, and the early hierarchy or classification of these organisms included kingdom, class, order, family, genus, and species, with species being the most specific category. In the future, we're going to see that additional categories are important as well. There's going to be one that we see later on in the chapter called domains, domain, and that's above kingdom. And then after the kingdom, so a little bit more specific than kingdom, let me narrow my pen, we are going to have a category called phylum, singular, or phyla, which is plural. So here's Linnaeus, the father of modern taxonomy. He actually is also credited to have invented the index card. He apparently used index cards to organize the ton of information he had related to categorizing plants and animals, which made it easy and efficient for him to organize information, change the categories easily. But he's also, on a negative note, regarded as having contributed to racism. So in addition to categorizing humans and plants and animals, it turns out he also categorized humans based on geography. And for his descriptions, he ended up perpetuating harmful stereotypes of people. So while he played a great role in his contributions to taxonomy of organisms and microorganisms, it's also problematic that he had a big role in the history of racism as well. As technology advanced, scientists created better and better systems of organizing or classifying organisms. And soon enough, scientists started getting interested in not only categorizing these different organisms, but also determining their relationships, specifically their evolutionary relationships. And these are known as phylogenies. If you draw this out and you can depict these relationships through a diagram, these are known as phylogenetic trees. So diagrams that show these evolutionary relationships between these organisms. To start off with, they were organized and connected through visible traits, like did they have the same number or types of limbs? Did they have hair or no hair? These days, though, because of the technology we have, we draw phylogenetic trees based on additional traits, such as their genes, their biochemical traits, or embryological data. And early in the 1700s, when Linnaeus was around, he only proposed two kingdoms at the time, animals and plants. About 100 years later, we have this German biologist and physician, Haeckel, who added two additional kingdoms. He added Protista, which are unicellular or single-celled organisms, and a little bit later he added Monera, which were single-celled organisms that did not have a nucleus like bacteria. Here we have a depiction of Haeckel's phylogenetic tree. It looks very complex. If we zoom in here at the bottom, we can see the plant and animal kingdom created by Linnaeus. Haeckel added Protista and Monera. And then over here we have this, which translates to the common root of all organisms. So at the bottom we have the root of, or the common ancestor of all organisms. And about 100 years after Haeckel, we have our American ecologist Whitaker, who proposed another kingdom. So now it's 5 kingdoms, the fungi kingdom. And he also said we should add another level above kingdoms. And he called these empires. Empires are sometimes also known as super kingdoms. And he proposed 2 super kingdoms, prokaryotes, like Monera. Remember, these had no nuclei, no nucleus. And eukaryotes. These, the ones listed here, all have a nucleus. So super kingdoms or empires are kind of the precursors to the domains I mentioned earlier, and we'll talk about that soon. But Whitaker's framework was used for many, many years. So here's the timeline for our different phylogenetic trees. We had Linnaeus and his animals and plants. Then remember Haeckel created 2 more kingdoms, protists and Monera, and then Whitaker added the fungi. So if we look at this check your understanding question from the book, or point from the book, we can see how our classification methods change with what we see, what we're able to see over time, and our technology available at the time. We started with Linnaeus and very simple 2 kingdoms. We had animals and plants. And then when Haeckel came around, we must have been able to see single-celled organisms because he added that new kingdom, protists, those unicellular organisms. And he also added the kingdom Monera. So we must have been able to see the difference between nucleated and non-nucleated organisms. And then later on, when Whitaker came about, he was also able to identify fungi as a separate group. The next part of our textbook jumps back to that clinical focus problem, and we have part 2 here, where Cora shows no signs of improvement after having been on antibiotics for a few days. Her labs come back and there are no bacteria or fungi in her CSF sample, so it can't be bacterial or fungal meningitis. And unfortunately, she has additional symptoms, so muscle stiffness spreading further, involuntary jerking, basically worsening cognitive symptoms as well. What kind of microorganisms do you think now might be causing Cora's symptoms? So keep that in mind. Early taxonomy or classification was based on visible traits, as we mentioned earlier, what you can see. But these can be subjective, right? They can be different depending on who is making the judgment call. If we were separating organisms based on size, for example, and we had 3 categories, small, medium, and large organisms, what I think is large, someone might say is medium to them, right? Luckily, technology these days, such as molecular genetics techniques we have, allow us to have and create more objective comparisons between these different species. And today, we often use techniques to look at DNA and RNA sequence or protein characteristics in order to create these phylogenetic trees, these trees of organisms based on their evolutionary relationships. Organisms that have more similar genetic sequences are going to be considered more closely related. And in the 70s, our American microbiologist Carl Woese and his colleague George Fox discovered that you can use ribosomal RNA sequences. Remember, ribosomal RNA is part of the structure of the ribosome, and we're going to see that in a later chapter. But if you look at the rRNA sequence of different organisms, you can categorize them fairly easily. He discovered, or they, Woese and Fox, discovered that archaea are a separate group from bacteria and eukaryotes. And archaea used to be called, or used to be known as archaebacteria. So you might have seen that in earlier studies of biology. Based on their findings that gene sequences coding for small subunit ribosomal RNA, or rRNA, of different organisms could be used to create phylogenetic trees, they proposed this three-domain system, which includes bacteria, archaea, and eukaryotes. So bacteria and archaea are prokaryotes, eukarya are in this eukaryotic category. And additional evidence showed that archaea are actually more closely related to eukaryotes than bacteria are. So it's similar to what I listed earlier, but we find that bacteria probably came first, followed by archaea, and then eukaryotes. These phylogenetic trees, instead of being called the trees of life, are sometimes called the webs of life. And this is because there is this interesting process we're going to learn more about later, known as horizontal gene transfer, that allows some organisms to pass genetic information to other organisms within the same generation. So imagine one bacterium can pass some of its DNA onto another bacterium. That makes it really difficult to determine how they're related evolutionarily. Here's a depiction of Woese and Fock's phylogenetic tree, and I kind of like this "you are here" sign for humans. Remember, there are three domains: bacteria, archaea, and eukaryotes. And this is created by looking at genetic sequences. Remember that we looked at ribosomal RNA sequences and how closely related organisms are based on their similarity in genetic sequence. The reason, or one of the reasons we use ribosomal RNA is because this RNA is considered a type of conserved gene. These are conserved genes. Oops, E right there. These are genes that have remained relatively unchanged throughout evolution. So this provides a pretty standardized way to determine the relationships between organisms. And down here we can see just a single line, right? A single line, which means that all of these, bacteria, archaea, and eukaryotes, evolved from a shared ancestor. And that takes us to our next check your understanding point. In the first question, they ask how we determine how closely two organisms are related. Remember, we look at DNA, RNA, or proteins, and the more similar their genetic sequences, especially remember in those conserved genes, the more closely related those organisms are probably going to be. And for the second one, why branches on the tree of life originate from a single trunk? I mentioned that just in the previous slide. Remember that that single trunk represents the common ancestor that is shared by all of those organisms. So based on this tree right here, phylogenetic tree, this suggests that bacteria, archaea, and eukaryotes all evolved from a shared ancestor, this trunk right here. Naming microbes is extremely important in our class. So let me put a star on this slide so that you remember to go back to this one or note it on your own notes. Naming was also invented by Linnaeus, and we still use his two-part naming system today. So this is known as binomial nomenclature or two-part naming system. And you'll see two words that we use to name organisms. So we've probably heard of Homo sapiens before in other courses. You might have heard of Escherichia coli, again, two names, Lactobacillus acidophilus, Bacillus anthracis. All of these are two-name organisms, right? Organisms we refer to using this two-name system. The first word in the name is the genus name, so that is always capitalized. The second word is the specific epithet, and that one is lowercase. Specific epithet sometimes is also referred to the species name, although the whole thing, Homo sapiens together, is also called the species. Notice that both of these names are italicized. So if you're typing this up, if you're typing something like a written report, and you're writing these names, they have to be italicized. Capitalize the genus name. Do not capitalize the specific epithet. If you're handwriting this, like when I write on the board, you should underline the names, right? Underline the names. Early microbe names were usually derived from Latin because that was the major language of the time when taxonomy was first developed and naming was happening. But these days you can see names that are Latin, Greek, English, and they can even reflect the traits of the organism, such as the example from our book, Haloquadratum walsby. The Haloquadratum, the genus name, comes from the characteristics of the microbe. So halo refers to salt, quadratum refers to the shape of the organism, and the second name, the specific epithet, actually comes from the discoverer of the organism, Walsby. After you write out both the genus name and the species name the first time, then and only then can you abbreviate the genus name. So in our class, make sure if you're writing a report, a paper, a summary, you always write out the genus name completely the first time. Then you can abbreviate for the rest of the paper. Okay, so again, let me star this because this is the second part of that slide, the naming slide. And the reason naming is important because it gives us this global consistency. We want to make sure we're talking about the same organism, especially in conferences, medical findings, publishing papers. Bergey's manuals, of which you can find some information derived from Bergey's manual in our Openstax textbook at the end, Appendix D, are comprehensive reference guides that are used by microbiologists and other scientists to either identify a microbe or classify it or to describe it. So this is especially applicable to prokaryotes, our bacteria and archaea. So in these manuals, you can see if certain bacteria, for example, if they have flagella, can they move? What size range are they? Do they have the ability to make something called endospores that we'll learn about later? You can look at their growth rates, what kind of requirements they have for growth. Are there temperature requirements, certain nutrients you have to have? What are their biochemical properties, genetics? So it's a really good standard reference, and we have a few of these in our lab that I'll show you in the future that you can use to, again, identify a prokaryote or classify them. There are two main types of Bergey's manual. On the left, we can see Bergey's Manual of Determinative Bacteriology. This one really focuses on identifying bacteria based on what you can see, their physical traits, as well as different lab tests. So this one is good for clinical and diagnostic labs. The one on the right, Bergey's Manual of Systematic Bacteriology, really focuses on taxonomy and looking at the evolutionary relationships between bacteria and archaea. And this one usually also has molecular data like the DNA, RNA, protein information for modern classification. Here's another checkpoint. So let's check our understanding. 1, what's binomial nomenclature and why is it useful? Remember, that was invented by Carl Linnaeus. So we name organisms using their genus and specific epithet names. Genus is capitalized, specific epithet is not, and we italicize when typing, underline when writing. This makes sure that every species has a very unique name, right? It only has a single name, so we can all ensure we're talking about the same organism. For the second one, explain why a resource like Bergey's Manual would be helpful in identifying a microorganism in a sample. Remember that it's a reference guide. A lot of microbes, when we look at them and when we start looking at them under the microscope in class, they're going to look the same. So Bergey's manuals, they give us information like biochemical tests. We'll learn about something called serological testing later, or genetic sequencing. They give us all this information and allow us to distinguish microbes even though they look very similar. Within one single species of microorganism, there can actually be several subtypes that we call strains. So this is more specific beyond that specific epithet. And these strains, even though they're in the same species, they can have very different traits, turning something that's typically harmless to very harmful. So E. coli is a great example of this, where many strains are harmless. We have them in our GI, in our gut, and they're actually beneficial because they help us digest certain nutrients. They help us produce certain nutrients. Others, like the strain you might have heard of, E. coli 0157H7, named for certain antigens, certain structures on these bacterial cells, are pathogenic. They are disease-causing, and they can actually cause severe foodborne illness. And you'll see there are symptoms and signs like abdominal cramps and diarrhea. It's linked to contaminated water, raw produce, and undercooked meat. So knowing specific strains is actually critical for diagnosis, treatment, and also food safety. All right, coming to the end of 1.2, we see our learning objectives again. Make sure you get your notes together and try to answer these in detail. When I talk about them, I just talk about these answers really briefly, so very superficially. Make sure you get your notes, answer these very quickly. Look at the first one, how microorganisms are classified and distinguished. We talked about taxonomy and naming systems. We can classify these organisms based on their physical traits, right? What we see under the microscope. But if they look similar, then we start relying on things like genetic testing or biochemical testing. Remember that each microbe or each organism has a unique, standardized two-part name, their genus and specific epithet. Looking at the second one, compare historical and current systems of taxonomy. Remember that we started with Linnaeus, which he really categorized animals and plants based on their physical traits, like what they could see. And then he created that hierarchical system. Later on, we have more advanced technology, so we're now able to sequence DNA, RNA, look at proteins, and start determining evolutionary relationships, phylogeny. Eventually, we started talking about the three-domain system, bacteria, archaea, and eukaryotes, developed by Woese and Fox. And then nowadays, we have Bergey's manuals that we look at a lot because we want to classify and discover new organisms based on their not only genetic sequence, but other biochemical traits. And that takes us to the end of part 2. In part 3, we're going to look at the different types of microorganisms out there.